Now, I request all the dignitaries to come forward for the lighting of flag. Dr. Divya to kindly assist.
Gourdas Choudhuri, let us now proceed. It's my pleasure to call upon our respected Vice Chancellor, Professor Shally Awasthi, to introduce our esteemed guests.
Respected Professor G. Choudhuri ji, who is the eminent father of gastroenterology in India. Our guest of honor, Dr. Samir, Mohindra, he is also a gastroenterologist to reckon with, tutored by Dr. G. Choudhuri, or I should say mentored by him. We have as a very special guest, my teacher and teacher of teachers, Professor Devika Nagman. We have with us indeed so many people here, but before I start addressing others, I bring an apology from the management. Our chairperson, vice chairperson, sent their good wishes and blessings to us today. They could not join us for certain personal commitment. So, I would now say that we are indeed happy to see all the faculty here, members of press and media, dean, vice chancellor, directors, heads of departments, students, and even staff here. It is indeed a momentous occasion for us at Dr. KNS Memorial Institute of Medical Sciences at Bodhisattva University to have hosted this event and to have such luminaries gracing this campus. Two of you for the first time today, and ma'am has been with us earlier. We look forward to seeing you again and again, blessing us with your knowledge. This institute began in 2012 under the vision and mission and blessings of late Dr. KNS MIMS sir. Today, we pay our silent tribute to him because this institution and organization is his dream, which is slowly fructifying and taking shape. Today's lecture will highlight the route from observations to patient care to community, and it will open new horizons for young generation to learn what research is and the nitty-gritties of research. Without taking any further time, I will introduce the eminent guests who are with us. Our chief guest, Dr. Gourdas Choudhuri. Dr. Choudhuri did his MBBS from JIPMER Pondicherry, MD from All India Institute of Medical Sciences, New Delhi, DM from All India Institute of Medical Sciences, New Delhi. Thereafter, he's got fellowship from JICA, from DAD, Germany, WHO, University of Sydney. In his academic career, he was faculty in Sanjay Gandhi Institute of Medical Sciences for 25 years, 10 years as professor and head of Department of Gastroenterology. He was the one who started, not in SDPGI, but in the entire country, EUS, that is endoscopic ultrasound in GI, and he started the biliary extracorporeal shock wave lithotripsy in the country. He has many publications and books. Currently, he is the Chairman Gastroenterology Fortis Memorial Research Institute. He has been Honorary Secretary of ISG, President of ISG, Founder Director GEM Program, and Director of GEM Program for 6 years. Dr. Choudhuri is known for his social work. He founded the Hope Initiative, which is health-oriented program-oriented education in 2004. Besides all this, his hobbies are games like cricket, hockey, tennis. He writes, and you can see his book Health Adda. And if you are interested, you can go and buy the book. It's on Amazon. I think it's being sold on Amazon. And with these words, we are very happy to have with us. Our guest of honor is Dr. Samir Mehra. He's currently Professor of Gastroenterology in SDPGIMS. He did his MBBS from LLR in Meerut, MD gold medalist from KGMU Lucknow, DM from SDPGIMS. Besides that, Dr. Choudhuri has been awarded gold medals in 8 subjects in MBBS, Pfizer Gold Medal, and Best MBBS Student of 1986 batch, Best Speaker Award UP Chapter Gastroenterology. He is a member of various eminent societies, and also he is the organizing secretary. He was the organizing secretary of ISG conference. Dr. Nag has been here with us earlier. No words can describe ma'am and tell about her glory. Ma'am has been my teacher, teacher of Samir Mohindra, teacher of Dr. Sudhir Kumar, and many of us from KGMC in this audience. Dr. Nag did his MBBS from KGMC in 62, MD in 65. She has had her academic training in neurology in USA and UK. She was faculty in KGMU from 73 to 1999, and she is the founder, head of Department of Gastroenterology, KGMU, and perhaps one of the earliest department of neurology in the country. With this small introduction to big people, I hand it over to Dr. Sada to continue with the program. Sada.
Thank you, ma'am. We are indeed fortunate to have such eminent personalities amongst us. As a token of appreciation, I would now request our Vice Chancellor, Dr. Shally Awasthi ma'am, to honor our chief guest, Professor Dr. Gourdas Choudhuri, with shawl and memento. I would also request our Dean, Dr. Sudhir Kumar, to present shawl and memento to our guest of honor, Dr. Samir Mohindra.
Thank you, sir. Now it's time to hear from our eminent guest of honor, Dr. Samir Mohindra, who is a renowned gastroenterologist. and is Professor in Department of Gastroenterology at SGPGI Lucknow.
First of all, I want to thank the management for making me a part of this function. It's an honor for me to introduce Dr. Gourdas Choudhuri, who has been my teacher, mentor, guide, not only in the field of gastroenterology, but in life also. And why I've taken out this piece of paper is because of time constraint. Because if I keep on speaking about him, it will take more than 1 hour. There is some repetition. Dr. Choudhuri has been an excellent academician. He started his journey from Jitpur, one of the prestigious medical college for MBBS at that time. And then he did his MD from AIMS, another premium institute. And then he entered into the DM program at AIMS. And mind you, there were only 2 seats for the whole country for gastroenterology at AIMS. So number 1 institute in the country and only 2 seats. And he was able to crack that exam. He was a very favorite. And at that time, basically the founder of gastroenterology in India has been Professor B.N. Tandon, and he was his mentor, and one of his most favorite students was Dr. Gourdas Choudhuri, what I came to know. I was not there at that time, but I know. And he was, if you pardon me sir, he was sent specifically to Indira Gandhi Institute of Medical Sciences, Patna, to start the program. At that time, a person belonging to Jamshedpur, with his education in cities like Pondicherry, Delhi, going all the way to Patna, just because his mentor asked him to do that job. It is amazing. And I know that he went by a train. At that time, there were very few trains. It used to get late. I mean, it's amazing that his mentor meant so much for him. It's a lesson to learn that your teacher should always remain a teacher and you should respect your teacher. At present, he's working as Chairman, Department of Gastroenterology at Fortis, Gurgaon. Ma'am, I've already told, he was the first person to start endoscopic ultrasound, ESWL. He has excellent publication, over 250, with a citation of over 6,000. And not only an academician, he basically is a very good administrator. He worked in SGPGI Department of Gastroenterology for more than 25 years, but out of which more than 10 years he worked as an HOD and he steered the department to greater heights. And I was fortunate to have his association since 1997. In addition to his being a very good gastroenterologist and a physician, another important aspect is that he's an excellent writer. And his column known as Atta is very sought after. And in Lucknow, if you ask anybody who reads Hindustan Times, his column is one of the most sought after. And people love his column, and I also started reading it. And in addition to that, he has written many books, not only in the field of gastroenterology, but in, for example, Health Under 10 Steps to Health. I think there's a recent book on gastroenterology and hepatology with one of my junior Piram, and it's an excellent book. You should all try to read it. Another important aspect is he's a very good social worker. See, when we do medicine, we restrict ourselves to the field of medicine. But in addition to medicine, gastroenterology, whichever field you are, you have some responsibility toward the society. And one of his biggest contributions, in addition to being a gastroenterologist, is his Hope Initiative. It basically looks at care of kids, whether it's their education, whether it's their health. They are doing amazing work. Mr. Negi is there, Mr. Rajesh is there. They have been associated for more than 20 years. And they are doing excellent work without making any noise. And a lot of kids are being benefited. And we should all give claps for that. So I know time is limited. So ma'am, I would not like to come between Dr. Choudhuri and the audience. So I once again thank you, sir, for being a mentor, guide to me, and I will always remember that. And I want to welcome my husband.
Thank you, Professor Mohindra. We are now at the most awaited part of the event. It's my privilege to invite our chief guest, Professor Dr. Gourdas Choudhuri, Chairman of Department of Gastrointestinal and Hepatobiliary Sciences at Fortis Memorial Research Institute, Gurgaon, to deliver the keynote address on the topic From Observation to Laboratory to Patients to Street, the Journey of Translation. Sir, we are honored to have you with us.
Thank you very much. Special thanks to Dr. Shally. It's a pleasure to meet Dr. Nag after a very long time, Professor Nag. And I have dear friends Samir, colleague, and Dr. Sudhir and Dr. Manish, the two deans. I've seen a lot of senior dignitaries here who I have interacted with during my stay in Lucknow and continue to do so. I'm a pure academician at heart, so I'll come to academics straight. May I just, before I begin, it's almost like a classroom, at what phase of life are you people in, most of you who are wearing white coats? Are you under? So you are doing your MBBS or you have finished your MBBS? Doing your MBBS. That's good. I mean, I just need to know at what level I need to pitch the talk. Oh, that's very good. It shows that the people who are undergraduates have more confidence. They raise their hands first. As you become more senior, the confidence levels dip. You see, you people raise your hands later. So if I ask, who are the teachers here, they will raise their hands the last. Putting them to embarrassment. So I'll begin straight with my talk, and why I want you to understand the talk is that, when you look at an issue, you can look into it in great detail, or you can look at the broad lessons which it has taught. Okay? So here now, for many of you who are undergraduates, I would want you to look at the broad lessons. For some of the postgraduates, a little bit more specific issues. But let me tell you, don't look upon it as a chapter in gastroenterology. It is an illustration of how translational research works. And for many people who are probably going to do research, wherever, be it in the faculty or residence, the time has gone when you could do individual great work. Nowadays, it's a time for collaboration. It's a time for working together in teams, taking ideas from each other, and only that will progress. I won't put any of you to any embarrassment, but if you look at the number of Nobel Prizes in medicine in the last 10 or 15 years, there's hardly ever been a single person who got it. It's been usually 3 or 4. Okay? And most of them have not worked in the same lab, but they have interacted with each other and learned from each other. Okay? Do you have a point here? So that's where I am working now. It's a very fancy hospital, Fortis Memorial Research Institute. This is my place after superannuation. ये बढ़ेगा This is very, very, I feel at home. This is like Lucknow. It's called Ulta Pradesh. But I love it. There must be humor in life, okay? So that's where I am at this point in life. And this is where I spent my 25 years where I learned much. Okay? How many of you have been to Sanjay Gandhi Institute? Some of you, all of you. Very wonderful. So I'll tell you how, and this cuts across any discipline in science, how an observation is a simple observation. But some people pursue an observation which is unusual. And the intelligence is required in noticing what is unusual. And if some people observe what is unusual, 1 out of 100 may decide to pursue and find out what was unusual about it. Most of us go through life, chalo yaar chalta hai. But you know how that single observation can make a difference. All of you have heard about Newton watching the apple fall from the tree. Again, people before him had seen it happen, people after him had seen it happen, but only it was Newton who said, why did it fall from the tree to the ground? So here I'm going to talk a little bit about something in hepatology, to talk and illustrate about what is called translational research, which means how one step from observation leads to a little bit of finding and then a little bit of translation across science in its cross-sectional areas to how it makes a dent in the community. Okay. So for undergraduates, look upon it more as an example. So this is how it all started. This gentleman, his name is Professor Kumar. He died a few years ago. But who do you think he was? I mean, apart from the fact that he got a Nobel Prize, he did. But what do you think was his specialty? Anybody? By the way, do you have enough chocolates? I love to distribute chocolates to people who give right answers. So at least volunteer to give answers. Anybody would like to guess? Since I'm a hepatologist, your first guess would be he was a hepatologist, right? Anybody thinks otherwise? Some murmurs?
Media.
Huh?
Media. Media.
Media?
Media. Bloomberg channel.
No, that Bloomberg is a different Bloomberg. You're talking about the TV channel. No, this was a different fellow. His name was, I'll tell you the exact name. He was basically a demographer. Demographer is a person who deals with different ethnic groups. You know, like if you look at UP Banyas and you look at Tambrams in Tamil Nadu, you know, people who look at populations. And there is a science behind it. Okay? So he was basically a population scientist. So what happened is that he was studying populations and it happened to be Australian aborigines. Okay? And you know, many ethnic groups have different types of genes. That time genes were not so much in vogue, but genetic markers. So he tried to put sera of some people of one group against another to see if there are antibodies and so on. And this is what he found, you know. So, sorry, I have a pointer here. So this is known as a line which indicates that there is a reaction between what is in this well and what is in this well. So it is an old-fashioned technique called gel diffusion technique. Auster-Looney's gel diffusion technique. Don't get into history, but the idea is if an antigen and antibody reacts over a period of time, it forms a line. Up to that much is clear, all of you? Yes. So when he saw this line, he thought that there is some antigen in these Australian aborigines which is specific to the genes or the tribe person. So he called it Australia antigen. Okay, remember that he was a demographer. He was not a virologist. Now after that, he did not give it up. So there were a lot of other people got into it. And then they found that it could be a genetic marker for Australian aborigines. But people who had that mark or antigen, they had a proneness for developing liver cirrhosis. Okay? And it went a bit further. So when people start exploring, they found, oh my God, there were people, Caucasians in Europe and America, many of them also had that antigen. But they had nothing to do with Australian tribes. So then they realized, okay, it may not be specific to the aborigine tribe of Australia, but to something else. Okay? So this is how the story evolved. And also people who had that antigen were more prone to developing liver cancer. So the whole thing shifted from Australian aborigine to something else which was causing liver disease. And this is the person, his name is Baruch Samuel Bloomberg. He thereafter became professor at the Fox's Institute in Philadelphia. And he got the Nobel Prize. And his Nobel Prize, by the way, occurred at a time when I was an undergraduate student. That was in 1976. I was in Chittor, Pondicherry. So this is what happened. And then they pursued further and realized that it was actually a virus. But the name had become so fixed, Australia antigen, that it went on for another 20-30 years as being called Australia antigen. So even now in Lucknow you find many reports coming as Australia antigen positive. Okay, that's the old story, but anyway, that's the history part. Now, if you put that under the microscope, now this is how evolution of science occurs. So there was an observation, but in that year was 1968-69. He got the Nobel Prize in 76. 76, there was not much by way of electron microscopy. Okay. So it evolved. So you see, science evolves in parallel. So you have science, the technology part evolving, the physics evolving, biology evolving, okay, and then when they come together, then you realize something is happening. So this is what they found under the electron microscope. So you had these virus-like particles, and there were a whole lot of these tubules, which now we know is what is called as the hepatitis B surface antigen. Okay, and this is the virus particle. Anyway, I'll go a little further. So as technology evolves, pictures get better. So how many of you have very advanced Apple cell phones? Anybody has got an Apple 14 or a 13? See, one of the ads nowadays is how clear your pictures can come with a camera. And resolution and perfection and catching stills. So as your technology improves, your ability to take pictures and understand becomes better, and this is exactly what happened with the hepatitis virus. And I'll now break it up a little bit more into the translational part of it. So once it was discovered that it's a virus which is possibly linked to the liver, there are different streams and groups of people. So you see three big groups here, according to the seating arrangement. So just imagine that there are some people who started looking at the virus alone. You may not be academically inclined, but as an example, so suppose one group of friends decide, let's go into the virus part and study it better. Some person might say, let's try to look at the transmission levels and prevention strategies. Okay? And some people decided, let's try to look at the immunology and how to develop a vaccine. So this is the same thing unfolded with COVID recently. And I think all of you from your age, I can make out that you survived COVID. You know how it all happened. Okay? It was a RNA virus. So there was a lot of debate on the television as to how it spreads. So there were people who got cranky about whether to take newspapers from the stand or not. Do you remember? Washing your hands, sanitizing the tables. So a lot of housewives spent a lot of time putting that antiviral on the dining table and the chairs and all that. But essentially it was spreading from the droplets, And then there was a huge amount of research as to how far the droplet carries. So the highest amount that I heard was 12 feet. And that's why COVID was such an effective part. Okay. So the principles remain the same. Now you see, in these three areas, how did we look at it? So a little bit more of science, and then it turned out that this was a DNA virus. So I'll give you a simple trick for undergraduates to remember. All the liver viruses, so you have A, B, C, D and E, 5 of them. Remember that all of them are RNA viruses except B. B is the only DNA virus. Okay? So this is how it all evolved. So this is what the picture looks like. This is what the incomplete DNA is like, the double-stranded DNA. It has got something else, the surface antigens, the soluble antigens and all that. And you do a little bit more. So I had this colleague of mine Rakesh Agarwal, who was very fond of looking at things in great detail. So they call it the open reading frames, So this is the template of the DNA which helps replication or production of antigens, or shall we say proteins. Okay. So these are the cycles which it is. So it becomes so complex. Now I'll go a little far. So after that, let's look at how to understand prevention and strategy. So here again, I'll ask a question to many of my undergraduate colleagues, postgraduates, if you are willing to answer the question. There was one particular infection which almost reduced the world population by one-fifth of the people died. And it came in waves. And do you know that was suspected to be an infective agent, but they could never see what the infective agent was till 100 years later. Can anybody guess which was?
Plate.
You are probably right, but you are right, very good. But the other one that I am referring to was the one which is called cholera. Okay? So if you people have not heard the name, it's called John's flu. And it's very interesting. If you are a good epidemiologist, you don't have to see the virus. You can plot it out by epidemiology. And what Snow did was that in the London epidemic, he just found out which are the parts of London, 4 parts, which had a similar water supply. And he could trace that the infection occurred only there. Okay, so that's the importance of epidemiology. How many of you are willing to become epidemiologists? Now after John Snow and after COVID, anybody interested in public health? No, I think all of you are interested in only clinical medicine. But public health is a fascinating area. I would encourage you. So now let's go a little further that how to prevent this infection. Now we know it's a virus. We learned a little bit about epidemiology, but not really fully. So this is the next part of epidemiology. Now who gets this infection? So they studied this without going through the virus. They found that people who had been getting blood transfusions and the blood was not adequately tested, they were at a high rate of getting it. So dialysis units where there was intravenous issues going on, the hepatitis B rates were high. Those who were using non-disposable needles and syringes, as it used to be in our days, they were pretty high. And now both these two have been more or less taken care of. And the commonest source of infection we see today in the community, when you get an incidentally positive HBsAg positive, is actually mother-to-child transmission. So these became the main methods of transmission, and they could find it out even without going to the virus. And these are some of the families which I had, and these are by the way from UP, and many of them come to me in the clinic. So if you do a family screening, you will find that from one index patient, you get the other people in the family to test themselves. You would find a whole lot of them have positivity, which means it is in the family, but you have only focused on that one patient. So you have touched the tip of the needle but missed the rest of it. Okay? Now let's look at the test and vaccine therapy. Today I'll make it short. I know some of you are already yawning, but I also have a selfish interest. I have to catch a flight. So let's look at what happened with the vaccines. So how would you make a vaccine? For example, anybody here, and this is general knowledge, it doesn't have to be a great super postgraduate to tell you, but any idea what were the approaches used for making the COVID vaccine? Anyone? And I'll promise that Shally will give you some chocolates, whosoever dares to answer. But this was all in the television. So anybody wants to take a guess? Everybody has heard of mRNA vaccine? Yeah, but that's the one which is way down at this point in time. But what were the other approaches used? Sorry? So the usual methods are these are infected particles which have been inactivated. And there are many methods, and that's the way traditionally vaccinology has gone. So they use that method. They use the mRNA method, which was a synthetic method. Okay, and they used the full particle method, which was a what was the Indian company? Hyderabad based. So there were different approaches used. The idea is that you have to get an antigen which you believe is the protective antigen or the antibody, and then you stimulate the antibody response using that antigen, and then once you get protective antibodies, you hope that you're good. Now you see, so many things happened with the COVID vaccine. So let's draw parallels. So the most effective vaccine was thought to be the mRNA vaccine. Okay. Why do you think that became so popular, apart from the fact that there were two American companies which were promoting it? One was the Pfizer and the other was the... Any idea? Why did that become so popular that many rich people from India flew all the way to England and America to get an mRNA vaccine? You know all that story, don't you? It was in the day press. What do you think the people spend their money doing because of that mRNA vaccine vis-a-vis the other vaccine, which was the Indian vaccine, which was a whole particle vaccine but inactivated vaccine? What could have been the concern? In a way, yes, but I'll tell you, the main concern was that when you actually make a vaccine from a viral particle, live viral particle, somebody will inject that idea that it is coming from a virus to कहीं पर कोई infection आपको ना लग जाए, ठीक है? And because mRNA vaccine was a synthetic vaccine, it conveyed a sense of assurance that there would be no chance of infection. It's totally synthetic. History has a very interesting story that unfortunately today the mRNA vaccine is considered to be one of the bad vaccines. Not in terms of protection, but in terms of the side effect profile. Whereas the Indian vaccine is supposed to have the best side effect profile, but the protection levels may be slightly less. Okay, so it's very interesting to look at how immunology works. It's tricky. So the most effective vaccine that was initially made, and this was published, by the way, in New England Journal of Medicine 1982. I remember the date because I was giving my MD exam at that time. And how would you give a vaccine for hepatitis B and follow up for a year and see if it is protected? Those days there were no antibody tests. So what they did is, there was a place in this world, it's called New York. And New York actually had a very high number of homosexuals. And homosexuals are very prone to developing all these infections, hepatitis B, HIV, and so on. So they invited homosexuals to come and get a vaccination done free with this vaccine, which was actually produced from the surface antigen in the serum of infected patients. And incidentally, that vaccine turned out to be very effective. And WHO was promoting that vaccine, and there was everything going for it. There was really no scientific reason, except some people created that fear that ultimately it is taken from infected patients. So, there is some other virus which you are missing. Now, but that's what happened. But having said that fear did creep in, and technology works in parallel. I just showed you how scanning electron microscopy came, and immunology came. So they had actually used these tubules. They had inactivated with a little bit of heat, and then they thought that it's not infective, and that's why the vaccine was used. And actually, till date, there was really no case of infection with the vaccine. But that fear came. Now you see, this is the effectivity of the vaccine. So the part of the country which had a very high rate of infection, but it was an educated country, it's in the storm or limelight right now. Any idea which is this country which is in the so-called crossroads or fight between China and America right now? Very good. Now, who said that? You deserve a chocolate. That you have to give some chocolates. So all the chocolate guys, what you do, you will come to Shally. I will arrange for the chocolates. Okay. Taiwan had a very interesting person called Dr. Beasley, and he was working on this new infection, and he started using this vaccine in Taiwan. And look at that year. So it started a little early. I told you this vaccine came somewhere, and then it was started. And over a period of time, can you believe it? Their rates of infection in the population dropped to almost as low as the Western Caucasian population. And that was a time when India was still struggling because we didn't have a vaccine program. And that's when Hope Initiative started. Okay? So, but that fear had come. So what do we do next? So whenever somebody comes out with a good scientific idea, you try to use that and see if it's better. So the issues or concerns were transmission of infections with the plasma-derived vaccines, whether the plasma proteins could be causing some side effects. How to get such large amounts of plasma from infected people? Because that meant you have to get a carrier, take huge amounts of blood, separate it out. I mean, it's ethically becomes a bit of a problem. How many people can you get? And there will always be a north-south divide because most of these infected people will be people from Africa and Australia, and the beneficiaries would be the people in Europe and America. So, there would be a So they came out with, so in 1982 it was the plasma-derived vaccine, and then they tried doing something called a recombinant HPV DNA vaccine. A story very similar to what Pfizer did with the messenger RNA vaccine. So I want all of you undergraduates that you should look at parallels. So I am giving an example from hepatitis, but you have to apply the principles to any other infection also. So these were the three people: Paul Berger, Herbert Boyer, and Stanley Cohen. I don't think any of you would have known the names, but they all got the Nobel Prize because of this technology called the recombinant DNA. Okay? And today, the vaccine which you get, which has become very cheap. Earlier on it was 500 rupees a dose, and now Rajesh and Negi give it to children at 50 rupees a dose or less. So what happened is that there has been very good amplification and production of this vaccine. It has become within the reach of a common man to the extent that our government has included it in the expanded program. Okay? And this is how the creation of the vaccine is, because you see this the technique of DNA, recombinant DNA, this is what you do. You take a little bit of the surface antigen ORF, the open reading thing of the DNA, and then you insert it into a rapidly multiplying cell. So the common cells used are either yeast cells in culture or certain bacteria which are in cultures. Okay? You can actually even put it in potatoes. And when it multiplies, it actually produces a huge amount of surface antigen protein without the viral part. And this is the vaccine which is now readily available, all of you, at a very affordable price, and this has changed the dynamics of one infection in the world. So next question that I'll ask to some of you, think of it if you are aspiring for a chocolate. Which are the infections which have been actually almost eradicated or very well controlled by vaccine? Very good. So smallpox, yes, it has been eradicated. It was there in my time. I have seen it. One of the main deaths of smallpox was actually Jamshedpur. That time it was in Bihar. And how the WHO campaign took place. They had a very effective vaccine. And when it comes to polio, now I'll pose the question to you. This is the homework. So the first polio vaccine that came was an injectable vaccine. And for that the gentleman got the Nobel Prize, by the way. It was a very effective vaccine. But then the people were a little averse to taking injections. So oral vaccine came. And now what you do is what is called the OPV, the oral polio vaccine, right? So if anybody tells one of the two names, gets a chocolate. All of you said somebody said salt. Very good, very good. Shally, quite a few chocolates. Quite a few. But it's very interesting that salt vaccine was the first vaccine that was an injectable vaccine taken from the whole virus. And Samir vaccine is actually an attenuated vaccine, which is actually the virus, live virus, but attenuated, which is an oral vaccine. And both of them are great, but salt got the Nobel Prize. So it's very interesting to learn a little bit about vaccinology. So this is how we use the vaccine for hepatitis B. So charity begins at home. We decided that let's make SGPGI hepatitis B free. So we invited not just nurses and doctors and others, but most important are our class 3, class 4 employees. And also let it open to their families. So that's why we went on maximum drives for hepatitis B vaccination, and fortunately things really worked very well. This was initially with WHO before we broke out into home separately. And this is how the treatment, so now those who had got infected, now you can't prevent or do anything with them with the vaccine. So the treatment part also started developing, and this is how the treatment was. I won't take you through that. So a whole lot of molecules came. Initially it was interferon, and then there were different molecules. We have seen them evolving over the 80s and 90s and 2000s. And at this point, the favorite drug is usually tilophobia, allopurinol, or antibiotic. So that part, don't get into it, that's a little technical. So now what happened is, there was a lot of understanding of the infection, the virus, the immunology. So the lab work was done. Now the question was how to take it to the field and get people benefited from it. So in those days when I was young, so this is what I used to do a lot of rallies. That is, for those of you in Lucknow, you know that's Asma Hussain. She is quite a fashion designer, and her grandmother was my patient and she has been my patient. So we did a lot of, this is R.K. Sharma, our director that time. So we did a whole lot of these programs with many of the school children, creating awareness. And that's how the message spreads. And these were huge halls full of people coming for a Sunday vaccination program, because they won't come until you have been able to use the media to impress upon them that this is a beneficial thing for which you need to come. See, the same story happened in COVID. Initially there were hesitation, but then when COVID vaccination camps started, there were huge crowds and big lines, because they realized it is for their benefit. And then, of course, we realized that we have understood hepatitis A, hepatitis B. D we will discuss some other time. It's a mutated virus. E is waterborne. And there was something else which was getting transmitted, but it wasn't B. And that brings us to the story of C, but that I will discuss some other time when I come back. Okay? So this is so far as translational research is concerned. I want all of you to take home the messages that you learned with one observation, and then looping in quite a few others from different disciplines who give their inputs. And if you are genuine scientists, any dissenting or different ideas shouldn't put you off. You should have an open mind to see is there value in it. And sometimes that benefit. If you think that, look, I am right, so anything which is sounding contradictory, maybe in the tone or in the character, and if you discard it, sometimes it's to your disadvantage. And different branches of science that work, and this is where we are at this point. I must put on record that most of my research in SGPJ, out of 250 publications, I think good about 50 of them were done in collaboration with CDRI, ITRC and FDRI. Because those days, we were in the very early stages and we didn't have much instruments. So Negi and I used to go, and IIT Kanpur. So we used to go and work with them from different branches. Those days my interest was in gallstones and bile. And I think at least 30 to 50 publications came because of collaborative research. Okay? Thank you very much for your patient hearing.
Thank you, sir, for the enlightening speech. I'm sure the message you have sent will inspire and mold the minds of younger generation present here today. To conclude the session, I would like to call upon our respected Dean, Professor Sudhir Kumar, to deliver a vote of thanks.
Esteemed Chief Guest Dr. Gourdas Choudhuri, Guest of Honor Dr. Samir, beloved teacher Professor Devikana, Vice Chancellor, Dean Academics, HODs, Directors, PG, undergraduates, and media. I'm very happy to be here today and proud to be presenting a vote of thanks to such a praiseworthy alumni of FCPI and another Dr. Mohindra also. These two people are really making a breakthrough in the branch of gastroenterology. Dr. Choudhuri, you have seen he had one of the best education, MBBS from Junior Pondicherry and the MBA BM from AIMS. And thereafter it was a very progressive journey. And today we had a good fortune having a glimpse of what he had been through. So you will find out that he is a very good orator, but the lecture was very lucid. He delineated very important features, but all with a very interesting course. So we are very thankful to you for the way of your interaction that had a very positive impact. And we hope you're going to see many more such type of presentation from you. Hepatitis B, you know, one of the dreaded virus and 100% mortality. This has been being tackled not by treatment, more of the prevention. That is why Dr. Choudhuri has emphasized that by having an observation, then we should have it across the discipline. The second thing which I think is very important, he has conveyed to you that you should have an open mind. You should not have something that whatever has been learned in medicine has been seen. So all of you can do wonders provided you are curious and you are research oriented and you learn the usual things what happens in the world. So I feel grateful, Dr. Choudhuri, that you are not only academic pursuits, but your social awareness, your campaigning to the public has made a great difference. Dr. Mohindra is also doing a very good job under the mentorship of Dr. Choudhuri, and I put my gratitude to him that he has come all the way and presented himself so that we can get inspiration and we can do well in future. Our beloved teachers who teach us, Dr. Devikana, whom I was a student, our principal student, our dean, with all our students. We have seen her in 73, 74, 75, 6, when she was a member of affairs. She was doing a very good job, and all of us KGMC, and then I feel very grateful and feel very proud to see her, and still she is very active, and she has got a special affinity with Dr. KNS group of hospitals. Still she is working with us. So I feel very thankful to very grateful for your kind presentation. Finally, students, PGs, faculty, we are very happy that you have been here, but I am sure you must enjoy this kind of interaction from a stalwart like a Choudhuri. So you should feel that all of you can try to find out the footprints and go ahead with zeal and enthusiasm. Thank you so much.
In the end, I would like to thank everyone present here for being a part of this thought-provoking session. Have a great day ahead.